Hippocampal and cortical neurons have been used extensively to study central nervous system neuronal polarization, axon dendrite outgrowth, and synapse formation and function. An advantage of culturing these neurons is that they readily polarize, forming distinctive axons and dendrites on a two-dimensional substrate at very low densities. This video demonstrates a technique to dissect, culture, and transfect embryonic mouse hippocampal and cortical neurons via nucleofection, which enables the expression of fluorescently tagged fusion proteins early in development, approximately 4 to 8 hours after plating. Expression of the fluorescently tagged proteins is maintained throughout the lifetime of the neuron for more than 2 months in culture. This enables the study of protein function during early developmental events such as axon outgrowth and later events such as synaptogenesis and synaptic plasticity. First, glial cells are isolated from cortical hemispheres of postnatal day 1 to 3 mouse pups and plated into flasks. After reaching 70 to 100% confluency, the glial cells are then replated on glass cover slips. Then, hippocampal or cortical neurons are isolated from E15.5 mice and transfected via nucleofection with plasmids encoding fluorescent fusion proteins such as red fluorescent protein. The transfected neurons are then plated in imaging chambers for short-term studies. For long-term cultures, glial cells on glass cover slips are inverted over hippocampal or cortical cultures to provide trophic support. Individual neurons can then be imaged to show the dynamic localization of fusion proteins throughout axons or dendrites of neurons.
Hi, I'm Eric Dent, an assistant professor of anatomy at the University of Wisconsin-Madison. Today, 3 of my undergraduate students, Chris Wieselman, Jason Ballweg, and Derek Lombard, are going to demonstrate a procedure for isolating, plating, and maintaining cultures of cortical and hippocampal neurons for short and long-term culture. These cultures can be used to study the dynamics of fluorescent fusion proteins throughout neuronal development, from initial neurite outgrowth through axon and dendrite development and synaptogenesis.
To prepare the chambers for imaging embryonic mouse, hippocampal, and cortical neurons, begin with 35 mm Petri dishes with 15 mm holes drilled in the bottom and all burrs removed. Melt a 3:1 paraffin petroleum jelly mixture in a conical tube by placing it in a boiling water bath. Then, use a small paintbrush to coat the underside of the dish around the hole. Be sure to stir the paraffin petroleum jelly mixture between chambers as it will separate. Place the cover slip over the hole. Once all of the dishes have been prepared, place them in an 80°C oven until the paraffin mixture is melted. This should take about 10 minutes. Then, remove dishes onto a flat surface and let the paraffin mixture set for at least 1 minute. Turn the dishes over and place them under a hood. Turn on the UV light for 30 minutes to sterilize both the insides of the covers and the bottoms of the chambers. Next, coat the chamber cover slips with poly-D-lysine for 1 hour. Then, thoroughly rinse the coated cover slips 3 to 5 times with sterile filtered deionized water to remove all traces of borate buffer. After the final rinse, use a vacuum to dry the chambers. These can be used immediately or stored in a tissue culture room for later use. Coated chambers should be used within 1 week of preparation. If long-term cultures will be prepared, cortical glial feeder layers will need to be started 2 to 3 weeks before continuing with the cortical or hippocampal dissections. Place the brains of 4 P1 to P3 mouse pups in a dish containing cold dissection medium and place it under the dissecting scope. Remove the olfactory bulbs, the 2 cerebral hemispheres, and the meninges. Transfer the hemispheres to a new dish containing no media. Using a clean, sterile razor blade, mince the cortices as fine as possible. Then, with a plastic pipette, transfer the chopped tissue to a 50 ml conical tube containing 12 ml of cold dissection medium. Add trypsin and DNase to final concentrations of 0.25% and 0.1% respectively. Incubate in a 37°C water bath for 10 minutes with intermittent swirling. Following the incubation, remove the tube from the water bath and thoroughly clean it with 70% ethanol before bringing it into the tissue culture hood. Pipette the cortical tissue up and down with a 10 ml pipette approximately 10 to 15 times, or until most of the chunks disappear. Return the tube to the water bath for another 10 minutes with intermittent swirling. Again, thoroughly clean the tube with 70% ethanol and bring it back to the tissue culture hood. Pipette the cortical tissue up and down as before, this time with a 5 ml pipette. Add 15 milliliters of warm glial medium and centrifuge at 1000 rpm for 10 minutes. Discard the supernatant and resuspend the pelleted cells in 20 milliliters of fresh glial medium. Use a hemocytometer to count the cells. Then, plate 5 to 7.5 million cells in 15 milliliters of glial medium per 75 cm squared flask. Place the cells in the 37 degrees Celsius incubator. After 1 day and every 2 to 3 subsequent days in culture, dislodge any loose cells by knocking the flask against the surface of the hood. Remove the medium along with any dislodged cells and replace it with 15 milliliters of fresh glial medium. Glia can be harvested after 1 to 2 weeks of growth in the flasks when they are about 70 to 100% confluent. To prepare individual cover slips coated with glia, begin by placing 6 nitric acid cleaned and sterilized 25 mm round cover slips in a 10 cm dish and, using a small paintbrush, place 3 dots of the 3:1 mixture of paraffin petroleum jelly onto each cover slip in a triangular pattern. Place 2 dots on the edges of the cover slip to anchor to the dish. Treat open dishes with UV light for 30 minutes. Coat the cover slips with poly-D-lysine for 1 hour. Then, wash extensively 3 to 5 times with sterile filtered deionized water and let dry. Remove the glial cells from the incubator, discard media, and wash with 5 milliliters of pre-warmed trypsin. Add 3 milliliters of trypsin to the flask and incubate for 1 minute at 37 degrees Celsius. Once the cells are detached, add 5 milliliters of glial medium to stop the trypsinization. Remove the glia from the flask by pipetting up and down 10 to 15 times, then transfer the media to a 15 milliliter conical tube. Centrifuge at 1000 rpm for 8 minutes. Following the centrifugation, resuspend the cells in 10 milliliters of glial medium. Then, after counting the cells, plate 5 times 10 to the 5th cells in 12.5 milliliters of glial medium per 10 cm dish containing the cover slips. Exchange the medium with fresh, pre-warmed glial medium every 2 to 3 days. The day before the neuron dissection, remove the glial medium and replace with serum-free medium. Use this glial-conditioned serum-free medium later when flooding cortical or hippocampal cultures. Era-C is added to a final concentration of 5 micromolar in order to prevent glial proliferation. Of the nucleofection reagents per transfection as indicated in the manufacturer's instructions. Then, warm the solution to room temperature. Place a dish containing E15.5 mouse brains in cold dissecting medium under a dissecting microscope. Use a bent tungsten needle to remove both neocortices. Then, using microforceps, remove the meninges and place the cortices into a new dish of cold dissection medium. The hippocampus and cortex are visible along the medial side of the cerebral hemisphere. With small iris scissors, carefully remove the hippocampus. Next, remove the remaining cortex. Repeat the dissection on brains from the entire litter. Place the hippocampi and cortices in separate 1.5 milliliter microfuge tubes filled with 1 milliliter of cold dissection medium. Place the tubes on ice. After dissecting all of the cortices and hippocampi, add 110 microliters of 2.5% trypsin to the microfuge tubes containing the tissue and place the tubes in a 37 degrees Celsius incubator for 20 minutes. Using a P1000 pipette, remove as much fluid from the tube as possible. Then, add 1 milliliter of plating medium to each tube and wash the tissue by gently inverting the microfuge tube. Repeat the wash twice. After the final wash, add 1 milliliter of plating medium. Use a P1000 pipette to triturate the chunks 15 times. Then, transfer the supernatant and cells to a new 15 milliliter conical tube containing 4 milliliters of plating medium, leaving any chunks that remain in the microfuge tube. Spin the 15 milliliter tube at 350 rpm for 7 minutes with the brake off. Following the centrifugation, discard the supernatant and add 100 microliters of pre-mixed room temperature nucleofection solution for each transfection. Pipette up and down 5 times to mix. Next, transfer 100 microliters of the nucleofection solution and cell mixture to each new microfuge tube containing an appropriate amount of DNA. 1 to 2 micrograms of DNA per transfection for long-term cultures will label less than 10% of the neurons in the culture, while 5 to 10 micrograms of DNA per transfection will result in a higher transfection efficiency for short-term culture. Add the cell suspension and DNA mix to an electroporation cuvette. Then, place the cuvette into the electroporator. For mouse CNS neurons, set the Nucleofector program to 0005. Working quickly, add 500 microliters of the pre-warmed and equilibrated plating medium to the cuvette, and transfer the cell solution to a new 1.5 milliliter microfuge tube. Add enough plating medium to bring the volume of each transfection to 1 milliliter. After counting the cells, plate 3 to 5 times 10 to the third cells per centimeter squared for young cultures, or 5 to 10 times 10 to the third cells per centimeter squared for long-term cultures. Generally, 5 times 10 to the fifth or 1 times 10 to the sixth cells are used per transfection. To prepare short-term cultures, 1 hour after plating, flood the 35 mm culture dishes with 2 milliliters of warm, carbon dioxide equilibrated serum-free media. Flooding the imaging chambers results in a very low serum content, less than 0.5%. Short-term cultures do not need to be cultured with a glial feeder layer and do not need to be refed. To prepare long-term cultures, add 1 milliliter of glial-conditioned serum-free medium. Remove a glia-covered cover slip containing 3 dots of paraffin petroleum jelly and invert it over the 15 mm hole in the 35 mm dish. Then add another 1 milliliter of medium. To feed the long-term cultures, replace 1/3 of the serum-free medium every 2 to 3 days. After 5 to 7 days in culture, cytosine arabinofuranoside, or ARA-C, is added to a final concentration of 5 micromolar in order to prevent glial proliferation. The following paired images of representative living hippocampal neurons show both a differential interference contrast image and a corresponding fluorescent micrograph. Each of these cells has been transfected with EGFP tubulin and DS-RED2 in PCAX vectors. During stage 1, neurons will initially attach and over a period of a day or so protrude sheet-like lamellipodia and finger-like filopodia around the periphery of the cell body. During stage 2, the initial protrusions will differentiate into several neurites emanating from the cell body, usually remaining about 30 to 50 microns in length. During stage 3, stochastically, one of the neurites starts to extend rapidly to become the axon. The other neurites remain short and are termed minor processes. Most hippocampal and cortical neurons progress to stage 3 by 2 to 3 days in culture. During stage 4, which occurs over the next week or two, minor processes thicken, lengthen, and branch to become the dendrites. Here, these are shown in green. Meanwhile, the axon thins as it continues to grow and branch. By 2 to 3 weeks in culture, dendrites differentiate by forming dendritic spines, small protuberances along the dendrites. Stage 5 neurons will vary in their shape and size, but most should undergo this series of developmental stages. Transfected neurons may not progress through these stages if overexpression of proteins disrupts neuronal development. Also, neurons may not progress through all stages of development if the cover slips are not cleaned properly, or the polylysine does not adhere properly. Glia can also proliferate extensively, especially if ARA-C is not used. This is a phase contrast image of a 2-week-old culture in which the glia have overgrown the culture. In the long-term cultures, changing the media too frequently or not frequently enough can also compromise the viability of the neurons. This is a phase contrast image of a culture in which most of the neurons have died, and the remaining neurons have shrunken cell bodies and beaded processes. For comparison, this is a phase contrast image of healthy hippocampal neurons at 2 weeks in culture. Note the larger cell bodies and lack of beading in the processes. Also, at this stage, the dendritic processes have thickened and there is an extensive filigree of axonal processes.
Once mastered, embryonic cortical and hippocampal dissection, transfection, and plating of entire litter of fetuses can be done in 2 hours if it is performed properly.
While attempting this procedure, it is important to remember that hippocampal and cortical neurons are very sensitive to mechanical disruption. Also, it is important to keep solutions as cold as possible during the isolation of neurons to keep metabolic activity low.
Following this procedure, other methods like live cell imaging and physiological recording of activity can be performed in order to answer additional questions, such as how fluorescent fusion proteins of interest segregate to different regions of the neuron, if protein localization is associated with changes in synaptic activity, or how the functions of these proteins is affected by different pharmacological treatments.